Hi, good morning, everyone. Boy, I hate that. More than 25 years. It's like, oh, you could do it. Yeah, anyway. So, before we get started, I did have to tweak this presentation just a hair because when I realized I was, it's a social housing symposium, so I do a little bit of a deeper dive into social housing, but I'm a mouse person from way back, and that's going to be my focus here. So if you are feeling left out at all the charismatic megafauna yesterday, and you really mostly work with mice, well, I'm your girl. So, okay, no disclosures, and the disclaimers is that Harvard's not responsible for this. I am. So, okay. As you may or may not know, you probably know because it's been everywhere in the news, the scientific news, we are, as scientists, as veterinarians, as biomedical researchers, we are facing a crisis in reproducibility. Or at least that's what some people think we're facing. There's definitely there's starting to be a backlash, as there is against every bit of received wisdom, is this really a real thing? But we can say that we are having a hard time. We are having a hard time reproducing work that other people do. And the reasons for that aren't entirely clear. But it's really affecting things like drug development. It's really affecting things, working towards human and animal cures for disease. Because we get promising results and then no one's ever able to duplicate them. So, where does that leave us, right? Now what? Where does that leave us? Well, it leaves us with really an ongoing attrition problem. We have models with no validity, and we have kind of stopgap measures. So we have reporting what we've done with the ARRIVE guidelines. We have planning what we want to do with the PREPARE guidelines. And I have to go on record as, in one presentation I gave, I kind of was poo-pooing the PREPARE guidelines a little bit like, oh, come on, we're scientists, we should know what we're doing. And my colleagues rightly sort of slapped me on the nose with a newspaper and said, hey, everybody uses checklists. The 747 pilot, before they start, they have a checklist. Everybody uses checklists. The surgeon makes sure they count all the instruments. And I was like, fine, okay, I'm wrong. So I'm wrong. Checklists are good. Everybody use PREPARE as well as ARRIVE. But what's missing here, right? What's missing? Let me ask you a question. What don't you know? So how did you find out that you didn't know that? So I was involved in a past life in a pinworm outbreak at a facility, and my faculty asked me questions that I was sort of like, they said, well, did we bring this from home? Like, no. Can we get this? No. But I didn't know that they didn't know those things because I knew them so well, having worked in lab animal medicine for so long with mice. I mean, this was just sort of internalized stuff for me that I didn't realize they didn't know. So let me make an analogy here, right? Most vets, we're like this shallow mountain pool. Okay? We have a lot of knowledge, but it's not necessarily very deep. Right? We know a little bit about a lot of things in many cases. Whereas the PhDs that we work with, they're like slot canyons. They know a whole lot about not very much, right? They are really deep dive into the things they're really deep dive into. And that's great. I mean, that's why we're complementary, because they can say like, I really want to do this thing in rats, and I really want to do this, and we can be like, oh, they don't have gallbladders. And they'll be like, oh, dang it, because we know those things. And the best example of this that I have is I love to tell this story on one of my colleagues. When I worked for Charles River, I took this colleague out to one of our facilities, and the colleague and I were touring the facility. And when we got to the hamster room, the folks working in the hamster room brought up this tub of hamsters for us to just sort of look at. They're gorgeous hamsters. They're big Syrian hamsters. They're fat. They're sleek. They're happy. No missing fur. Everybody's eyes are bright, and they're looking around. And my colleague turns to me and goes, oh! And I said, what? And they said, where are their tails? And and I said, oh bless, okay, no, they don't have tails. And my colleague got super defensive and said, well, I know a lot about the things I know a lot about. And I'm like, yeah, exactly. And so they hadn't worked with hamsters in their career, so they didn't know that this was normal. They thought their first instinct was that this was some horrible welfare issue, that all these hamsters had chewed each other's tails off, because that's what they knew. That was the lens through which they were seeing it. So each of us has our strengths, each of us has the reasons we've chosen to do the things that we do, and it does help both sides understand if you sort of think about our knowledge as not as complementary in this way. So how do we know things? We know things through, this is epistemology, right? The theory of knowledge, especially with regard to its methods, validity, and scope. So epistemology seeks to answer the questions, how do we know things? How do we know that we know things? And how do we know that we don't know things? So we are investigating our justified belief versus opinion. So let's look at it in a slightly different way. Okay, so if we divide knowledge into sort of 4 quadrants, we know what we know, right? We know these things. Unfortunately, there's always a subset of things that we know that are going to turn out to be wrong. So that definitely counts. We know these things for sure, but oops, they're wrong. Then we have a slightly larger subset, the things you don't know. I don't know what the dose of ketamine for a non-human primate is. No clue. I wouldn't know how to formulate a dairy ration. No idea. But I know I don't know these things, and I know I can go to colleagues, and I know I can go to books, I can ask these questions. And then finally, by far the largest knowledge quadrant, if you will, is what we don't know. And these are the unknown unknowns. So we want to, in science, avoid the trap of failing to recognize these things. So we don't know them, so how do we do this? What we do is we keep an open mind, like, hey, I didn't know that. Now I know that, I can make different decisions going forward. And we acknowledge that what we don't know is by far the largest body of knowledge out there. So bringing that around to animal modeling, all of our models are imperfect, okay? We know that, that's a known. But what matters is how we address that imperfection. So we have three measures of reliability that we sort of look at, or 3 measures of perfection of a model, right? We have validity versus reliability versus reproducibility. And so any measure that I'm taking can be perfectly reliable, so always giving the same value under different conditions, that's great. And the experiment can also be perfectly reproducible, so given the same results when repeated, okay? But the validity of that experiment, the validity of that measure can still be rubbish. So validity is the degree to which a measure means what it's supposed to mean. And one of the best examples of this is, of course, phrenology. Everybody knows what phrenology is? Okay. It's the study of the bumps on your head. So this was a science in the mid to late 1800s where people would go around and they would touch your scalp and they would feel the different bumps on your head and they would measure them and, like, oh, she has a big bump in the maternal spot. She's obviously a very good mother. Or, oh, her criminality bump's a little big. Anyway, so 10 people could measure the different bumps on my head, the shape of my skull, and probably get close to the same answer. So phrenology has good reproducibility. The bumps on my head aren't going to move between times they're being measured. I mean, in most cases, right? So it's got great reliability. But what's the validity? What's the validity of this? Are these bumps going to predict my memory or my mothering skills or whether or not I'm going to commit a petty crime? No, they're not. And this is also to remind you that even rubbish science can have journals. So, okay, back to we don't know what we don't know. What's missing from using the animal models in the way we do? So what's missing, what tends to be missing for a lot of folks is the acknowledgement of what we're leaving out every time we choose a model or experiment or an assay. there's not a perfect model. You can't control everything, but you have to sort of be aware and acknowledge of the things you're leaving out and how this affects how your model works. So some of these you're going to know, and some of these are going to be unknown. So when we, Joe and Brianna and I sort of came up with the idea, the field of theory-epistemology, we came up with 6 questions to ask yourself, to ask your scientists, to ask the people who are working with the animal models. But the basis of them is, what did you choose to ignore in this experiment? And it's okay. They're models. They're not perfect. They're not, we accept their imperfections, but the more knowledge we have about the imperfections we're accepting, the better the models are going to be, the more reproducibility and more importantly, translatability we're going to get. So these questions are not fancy or difficult. You don't need to know anything to ask the question, but it may be a challenge to really answer these questions. And if you take home nothing else from this talk, if we stipulate to ourselves and to the outside world that there's value in animal research, and if we're going to be stewards of using this limited resource of animal experimentation, then it's really on us to be sure that we are using this resource in the proper fashion with the most downstream usefulness, okay? So here are 6 questions to consider. We're going to go through them briefly one by one with a focus on animal well-being sort of towards the end there. So what features of model biology are ignored? So again, you have your person who's developed a perfect experiment in rats, except for the fact that rats don't have gallbladders. What features of human biology are ignored? What features of the measures you're using are ignored? What features of background methodology and husbandry of the model are being ignored? What animal well-being issues are being ignored? And what principles of experimental design and statistics are being ignored? And that's a huge one. So we ignore model biology. We ignore it all the time. We ignore it through ignorance of species differences. We ignore it through managing only what we can monitor. So we can monitor for these diseases, these infectious agents, these are the ones we monitor for. And model biology is also ignored through ignoring experimentally induced changes in unintended aspects of model biology. To dive a little deeper into one particular question there, we ignore model biology through ignorance of species difference. The mouse's humble preputial gland. Okay, it's a paired sebaceous gland located on the lower abdomen, if you're not familiar with mice, and it varies in appearance based on hormones, behavior, and bacterial colonization. It's not a teratoma. It's not a squamous cell carcinoma. But if you don't know, if you've not dissected mice, if you don't have access to a veterinarian or a veterinary pathologist, or you don't listen to those people, then it's sure a weird-looking thing stuck there in the abdomen that you have no idea what it does and what it's for. And so people have written them up in the published literature, and it's gone through peer review, that these are paired squamous cell carcinomas. That's an unusual interpretation of that, but okay. So it's things like this where you have ignorance of your model's biology and you are just casting about for some sort of phenotype that obviously this must be a phenotype because it's pretty weird and I've not seen it before. So what features of human biology are ignored? So if you are modeling a human condition, what part of the human biology are you ignoring when you're trying to make this model fit this mold? So we ignore human biology through ignorance of human diagnostic criteria. Can you really model autism in an animal without a theory of mind? You can model certain behaviors perhaps associated with autism, but careful there. Same with depression. Human biology is ignored because human symptoms and a symptom is different from a sign because a symptom is self-reported and a sign is seen by others. Human symptoms are considered unmeasurable, which is absolutely not the case. There are plenty of scales and measures out there that humans apply to other humans. Human biology is ignored because doing something is perceived as better than doing nothing. And we also ignore human biology through sheer reductionism. A human is a mammal, a mouse is a mammal, therefore, it doesn't quite work that way. So often what we have when human biology is ignored because doing something is perceived as better than doing nothing is we have really highly motivated scientists. They really want to fix this problem. They've been perhaps affected by it personally. They have seen the suffering that this human illness or condition wreaks upon their patient population. So they're highly motivated to help this human patient population, but what can sometimes be difficult is that there is at times a difficulty in acknowledging that sometimes we need to move up the phylogenetic scale, not down, to better model these conditions. So, that's where we get the any model's better than no model, right? Because it's doing something, it's something. And instead, maybe these scientists should be recruiting patient populations. Maybe they should be working with MDs in the clinics rather than trying to model something in a way that's just really never going to be translatable. So what features of the measures that you're using are ignored? Okay, so features of the measure are ignored just through sheer ignorance of the discipline. So you read something, you think, well, that might be a good assay for the thing that I'm going to do, and you try it out without having the background knowledge behind you of the things that you need to know to use the assay or the measure accurately. You can also ignore evidence that your measures are flawed, and you can ignore sensitivity versus specificity with respect to the research question. So a lot of times it's, again, those well-meaning scientists. You can be a geneticist and do behavioral phenotyping, but you can't be an ethologist and get a slot in a genetics faculty. No, they don't play that way. Even though the geneticists are borrowing measures from the ethologists that they don't necessarily have the deep dive into the background to understand their proper use and their limitations, most importantly, their limitations. So respect the specialties and measures that you want to use, and remember, science should be collaborative. That's the best part, right? It's like, hey, I want to do this thing. I don't know enough about this thing, but Bob over there in animal science does. Let's go collaborate with Bob. And everyone wins that way when science is collaborative. Okay, so what features of background, methodology, and husbandry are ignored? We ignore some of these features because it's how it's always been done, and if you change things, the model stops working. Experimental background is ignored because of the one-size-fits-all industrialization of animal facilities. So we monitor what we can measure, and the easier we make it for us to measure things, the more tightly they're monitored, and this kind of reductionism has led to some fairly interesting experimental backlash in things like the people using the wild mice and finding that, oh, their immune system is much more normal than the immune system of these super clean mice that all the immunologists have wanted for so long. And experimental background is ignored through seeing animals as tools, not as patients. So that's a big one. So experiments happen in time and space. Right? They don't just exist, they don't just exist out of the space-time continuum. They have a place where they happen and a time where they happen. And there's a support structure around every experiment, and despite our best efforts at standardization, this support structure is a living thing comprised of many moving parts. We had some discussions yesterday about how caretakers affect the behavior of animals and how they, and so these caretakers don't, there's not one animal caretaker who's been working since 1903 with mice, right? So everything changes through time, and the biggest recent example of this is certainly the microbiome, and we're finding out how much the microbiome can have an influence on behavior, immunology, infectious disease work, all sorts of things. So the other thing is that if the model doesn't work when things change, was it a good model? The other thing, though, is that when things change and the model doesn't work, that's a new avenue of scientific inquiry. Why? Why did that happen? Let's see if we can figure it out. Now we didn't know that factor affected our mice and our model this way, and now we have a new line of inquiry that we can work on and figure out why that happened. Okay, so what animal well-being issues are ignored? We ignore animal well-being issues because we are ignorant of human well-being effects on health outcomes. You guys have all seen the studies about cancer patients, especially breast cancer patients, and survival and social networks. Things like warming blankets after surgery. These are becoming more standard in at least mouse and rat surgery than they ever used to be. But we need to think about the sort of the overall holistic environment of the animals, social support, things like that. Animal well-being issues are ignored through mistaking absence of evidence with evidence of absence. And then, that's a repeat there, that we ignore them by treating the animal as a tool, a little furry test tube, not a patient. So animal well-being issues are ignored through ignorance of human well-being effects on health outcome. So human illnesses don't happen, again, in a vacuum. And when we try to use animals, the way that we keep animals, when we try to use them as models for disease, human illnesses don't happen solely to a cohort of very closely related 45-year-old virgin men living only in a village in England who happen to live in solitary confinement, and a grizzly bear comes in every 2 weeks and cleans their house and deletes all their social media. Doesn't happen that way, right? But that's what we're modeling when we try to do these things to our mice and rats, rather than using more diversity, treating them like human patients, and using our statistical tools to eliminate sources of noise in our experiments and better find the background effects that we thought were lost. So what matters to people? Well, being warm, being safe, having a friend, having good food to eat, and a nice place to sleep. And that's just the basic stuff, right? If you're going to study cognition or dementia or learning in animals, then you have to think about other things that affect those factors in humans as well. So, social support, sleep, all these other things that we know affect how these outcomes work in humans. So what else affects animal well-being on study? Well, stress, of course. But stress is so broad, right? Is it distress or is it eustress? Anybody out here with children would probably say that it vacillates between those two states depending on the attitude of the child that day, right? Do you have control over the stressor or not? Is the loud noise going to happen is that drilling, that air hammer, going to happen again? I don't know. I'm a little stressed by that. If I knew that the air hammer was going to happen again, I'd look at my watch and say, okay, it's time to stop now. But I would feel better because I had control over the situation, or at least the illusion of control. And then are we using mitigation strategies for the stresses the unavoidable stresses that our animals are under, or are we either deliberately or inadvertently exacerbating these stresses? So let's talk about mice, because I love mice. They're my favorite animals. How do lab mice live? Well, they have very limited opportunities for free movement. They have little to no control of their immediate environment in general. They have a consistent temperature, humidity, and light, so both intensity of these things and duration. Their home range or den is disturbed regularly by an apex predator. And they're forced into social interaction with their conspecifics that they may not necessarily want or need. They have free access to highly caloric, relatively unchanging food, and they have genetic homogeneity across the world. But they do, lest you think the life of a lab mouse is just, horrible. They do also have freedom from disease, cold, heat, hunger, thirst, predation. So they have some benefits there, right? But they're very different from wild mice. They have free movement in 3 dimensions through their environment. They have control of their immediate environment to a certain extent. If it's too hot, they dig a burrow. If it's too warm, maybe they go, dabble in the stream. They have variation in temperature, humidity, and light as the seasons evolve. And their home range or den is still disturbed regularly by apex predators, not just one apex predator. They've got many predators because mice are nature's Doritos. They're just always there for a snack for anybody who comes along. And many of them have a choice in social interactions with conspecifics. So they can choose to, the females can choose to migrate from their natal territory into the territory of a new male. Or they can choose to stay with dad, or they can choose to kind of go it alone. But like bachelor males who can't have a territory, they don't have a lot of choice. But most of them have a choice. They have to work for access to changing food sources. There's not just one invariable food source available 24/7. Sometimes they eat lichens, sometimes they eat, bugs, sometimes they eat seeds, whatever, berries. I mean, they have to work for it, whatever it is. And there's genetic variation across locations. So, one really interesting thing though, and the reason that the mouse is such a great model is its phenotypic and genetic plasticity. So if you take these mice and you put them into a more naturalistic environment, and I would highly encourage you to go see the work of Wayne Potts, because it's super neat. He's taken these lab mice, put them in colonies in a more semi-naturalistic enclosure, and compounds or treatments that weren't showing effects in laboratory cages now show effects that are robust when the animals have to do more things. So when he has them in these semi-natural environments, you see all sorts of stuff that you didn't see when you tested these same things in lab environments. So I hope that this becomes a more popular way to do drug development and drug toxicity testing, because I think that we would see a lot of different things. All right, so what do we do that stresses mice and rats? And you're going to see that little asterisk after stress every time, because it's maybe, because it's not always a distressor, sometimes it's a eustressor. But these are the things that I'm going to talk about in a little bit more detail today. So what do we do that stresses mice or rats? Keep the room too cold, for sure, that's a stressor, right? Well, maybe what's too cold? I mean, what I think is pleasant, you might think is horribly hot or horribly cold. So, and mice have these individual variations as well, depending on life stage and other issues. And of course, it's obvious that it's very stressful to be singly housed if you're a mouse. Is it? Anyway. Okay, so keeping the room too cold is probably, that's a real thing, right? We know that when we keep animals closer to thermoneutrality, and humans tend to seek out environments that are closer to thermoneutrality, we know that mice are stressed in typical laboratory housing temperatures. It's too cold for them. And if you don't give them nesting material, you don't give them any way to control that cold or ameliorate that cold stress. And that cold stress has basic effects on their biology. Things like 1.5 times more food consumption, things like tumors growing a lot more slowly because they are using their energy towards staying warm and staying alive rather than growing a tumor like you want them to. So it can also suppress endogenous immune responses and severity of graft versus host disease. So there's a lot of reasons to keep your mice at a more thermoneutral temperature. But, I mean, that's really the thing, though, is that there's not one right temperature for mice. The little guys there, they're basically ectotherms. They need their mom to sit on them and be in a nest and keep warm. But the mom there to the right is, when she's lactating, she's wearing a jacket, like a full-body jacket of highly metabolically active tissue, which is making her super warm. So she doesn't want to be in that nest with the pups because she's like, oh. And so she goes outside, and this is some work by Kroll, who showed that when he shaved the backs of these lactating female mice to let them blow off more heat, they had pups that were like a gram or two larger at weaning because they could produce more milk because they could release the heat burden that this milk production provided or gave them. So it's not really practical in most cases to nair all your lactating female mice, but you can do something similar by keeping your room temperature at something that's comfortable for you to work in, something that's comfortable for mama mouse to take a step out of the nest and, have a breather, but give the mice plenty of nesting material so that the babies can be tucked away and stay warm, or the old gentleman living in the cage can be tucked away and stay warm and not have to use so much energy to just try and, keep homeostasis. So the other thing you need to know is that animals can be social in many different ways, all right? So if we think about sociality, so we have solitary animals that just have single interactions, okay? So like a tiger. Tiger comes together to mate, rears some cubs, everybody goes, they have their own territory, that's how they live. Then we have animals that have aggregations. So I think these are prairie grouse coming together on a lecking ground. So they are doing their mating thing within sight of a whole bunch of other prairie grouse, and they're having interactions, but their interactions are mostly about competition. You know, I'm the best prairie grouse, I boom the loudest, ladies, come see me. There's not very much cooperation there, other than a lot of prairie grouse in one place reduces the risk to each individual prairie grouse from predation, right? I mean, if you're the one prairie grouse, one predator versus 200 prairie grouse, one predator, okay, that's gonna work out better for you. Then we have animals that are social in schools. So they come together for these interactions. There's a little bit of competition, but there's also a little bit of cooperation and support, again, against predators. If there's 10,000 fish in a school, a shark may get 50 of them, but he's not gonna get all 10,000, and so they're cooperating in that sort of meta overarching sense. And then, so these are all sort of interactive behaviors that these animals have with each other. But then if you look at packs and colonies, they have relationships, okay? So in this case, in a pack, there's cooperation, there's support of individual animals, and in the furthest outliers from solitary animals, we have animals that live in colonies. There's no competition between animals, or very, very little, and there's a ton of cooperation and a ton of support, like bees. So where do mice fit in this spectrum? Well, if you think about it, you can argue that they fit in almost anywhere. Okay, so they can be happy as solitary animals. They can also be happy in something close to a colony if you think about the density of mice in the walls of your house. Is nothing like the density of mice living in a wheat field in Australia, okay? So, I mean, and the fact that we can force them into the small cages that we use, again, they're acting like colonies, they're acting almost cooperatively in this fashion. But what you have to remember is that social rodents are gonna socialize, okay? Great, right? Socialization, that's wonderful. Everything's great. But not all social interactions are happy or benign. And so that's something else we have to remember, okay? So when we ask the question, the regulatory question we always ask is, are mice social? Are mice social? Must they have a companion? Well, from an enrichment standpoint, in terms of using social housing as an enrichment, we have psychological stress and risk of injury, of course, because of the potential agonistic interactions between these animals, both territorial and dominance related. But we also have the opportunity for control, theoretically. I mean, in some cases, if we let mice choose their companions, they do a lot better with those companions, both breeding and sociability. So the right question is not, are mice social? The right question is there evidence for benefits of social support? Yes. Followed by, if so, yes, can we mitigate the costs of the inevitable conflict? Because there's going to be some sort of conflict. So being social isn't enough. We have to give them the right environment for them to be social in the right way. And there's a whole, I could segue into another hour on mouse behavior here, but I'll stop. So, but just to look at some relatively recent publications, Capel concluded that whether group or single housing is better or less worse is context dependent, of course, depending on strain, age, social position, life experiences, and housing and husbandry protocols. If you have mice that you have housed together and they're fighting, you can separate, like if you have 5 mice, you can separate these mice into groups of 2 and 3, for example, or 2 and 2 if you have 4 mice. And the mice figure it out. You don't have to wind up with 5 singly housed mice from a cage of fighting mice if you just give them a little bit of time. A compatible partner for a male mouse, even a castrated male mouse, isn't going to work out well. The intact male mouse is going to really have some problems with that. But an ovariectomized female will work out okay if you want to go that route, if you need to go that route to provide your singly housed male mice with companionship, but you don't want any babies, that may be the way to go. And the people who wrote this were at a pharma, so I mean, they might have some resources that the rest of us don't. And then in this very, very recent paper from Malati, that it's like May of 2019, pair housing is great. Pair housing is great for black 6 mice, but they separated them at multiple time points throughout adolescence and adulthood, and the mice were able to adapt to single housing and did not have significant markers of stress. So you can singly house mice if you need to. So, what else do we do that stresses mice or rats? We house them in individually ventilated cages. Maybe it's stressful, or maybe we house them in individually ventilated cages. At least the number of disturbances are decreased by IVC caging, because we definitely know that cleaning their cages disturbs mice. And housing them on wire, absolutely problematic, and this paper is great, the one that I have at the bottom of the page there, that shows that animals really don't habituate to metabolism caging, and so you should keep that in the forefront of your mind. If you have to use metabolism caging and evaluate results from that metabolism caging, you are evaluating results from a profoundly stressed animal. So mice find puffs of air aversive, so consistent results finding that mice like air coming in from the top, not at their faces. They don't like high flow rates, but if you give them nesting material, again, that control over their environment, they're okay with it. Reclaimed wood pulp bedding didn't control ammonia levels, so ammonia levels can be a problem if you try to lower the ventilation rates in individually ventilated housing. This is a small study that found that there's no welfare implications in going 21 days between cage changes in individually ventilated cages, but post-cleaning aggression, Pascal Van Loo, that's like, we all know this, we've all seen this, and she makes recommendations on number of mice in a standard shoebox cage based on this work. They drank more water, that was pretty much the only thing they found in Customopolis, and animals were more aggressive in in high flow IVCs than in static, so they were more aggressive towards each other in IVCs. What else do? We wake them up in the middle of their sleep cycle, right? Mice and rats are nocturnal, and we are in there cleaning cages and doing our work when it's convenient for us, a diurnal mammal. And so it's the equivalent of, housekeeping at 3 a.m. for them. And they're like, whoa. So, and we expose them to noise. But when you consider noise, again, you have to consider that things that we can hear, mice maybe can't hear, and lots of things that we can't hear, mice definitely hear. And again, there may be more benefits to individually ventilated caging there in terms of the provision of background white noise that keeps them from having problems with different frequencies. But they can also be a source of problems if they start making noises in those frequencies. There's a whole huge body of work on light at night in its effects on animals, so just sort of keep that in mind. Light at night changes behavior, including dim light. It can increase anxiety in mice. Cage changing disrupts sleep, although there's a recent publication by Brianna Gaskell that shows that mice do okay with this fragmented sleep, that they probably don't need a solid 8 hours a kip every night for them to be good mice, that this is probably how they live a little bit in the wild. Corn cob is not ideal, and we're working on a review paper on that right now. Routine cage management and procedures stress mice, and females are always seemingly more susceptible to these levels of stress, so good to keep in mind. Noise. Rats prefer silence in the animal room. They don't necessarily want to hear Swedish pop, nor do they want to hear metal or even classical music, although if you have to play something, classical music's better than nothing. Better than any other kind of music. Routine husbandry techniques are too loud, if you think about it, and they can hear vacuum, so the clanging and banging and stuff that we do in our animal rooms, definitely the mice can hear. And a typical laboratory animal environment is about 80 decibels, with peaks 30 to 40 decibels louder, and a decibel is a log scale measure, so that's good to remember. At 120 decibels, so if you're at 80 decibels and you have peaks at 120, you're going to see permanent hearing damage both in yourself and your animals. And at 85 decibels, you're going to have reproductive effects. So we would do well to be sure that we can mitigate sound pressure in our animal rooms, both sound that we can hear for our own OSHA compliance and further hearing health down the road, and for the animals and their behavior and reproduction as well. Some sounds that bug us with construction, mice can't hear, so that apparently that air hammer that we dislike so much, mice are like, what? So, and then Turner, it's a great review of laboratory animals, strain differences, the hearing, where the hearing ranges are for animals, so. What else do? We house animals far off the ground. So if you're a mouse and something's eating you, what happens? You are lifted up and you are taken away, whether that's into the mouth of a fox or into the claws of a hawk. So mice definitely find high, brightly lit spaces quite aversive, and this is the basis for a lot of the fear and anxiety tests that we do in mice. And we house them where it's busy. So mice and rats are 3 to 5 times more sensitive to motion, ambient motion in the environment than we are. So things that wouldn't bother you might be definitely triggering some behavior in mice that we wouldn't like to see. So there are definite rack and room effects. In this study, shelf height made animals more anxious, so the higher up you put them, the more anxious they were. Rack height can also stress the animals and makes them resistant to diabetes, so resistant to the condition you're trying to induce. There's a large body of work by Sylvie Cloutier, but this is one of her papers where she showed that there was no effect of height in stress on rats, but that rats didn't want to be visible to humans, so that if you gave them a choice between a clear cage and a semi-opaque cage, whenever the light cycle was on or humans were in the room, they went into the semi-opaque cage, but when there were no humans in the room, they hung out in the open cage and looked at the other rats around the room. So when given a choice, they were like, yeah, I'm gonna go away from the predator and hide for a little while, maybe I'll be safe. So the quick answer to this question, are there local room effects, is yes. And in tox studies, I guess they used to organize the animals so that there were, all the high dose are here and the medium dose are here and the low dose are here. And then they found that there were these definite significant room and rack effects where animals were getting tumors at different frequencies, not necessarily based on the compound, but more based on the stress of their particular housing. And this is one of our papers where we found one of the few significant, we tested a lot of things in this paper, and we found that many ways to make mice fight more. So if you want your mice to fight, let me know and I'll tell you how. But one of the more significant findings that we had in this paper was the room that we were working in, the racks were arranged facing each other. And we had animals facing each other like this. So the animals that were facing this way that could see the door that was on this side of the room fought more than the animals were facing this way where they just saw a blank wall or they saw the other animals. It didn't trigger them to fight as much as what we think was the motion of the door and the workers in the room. So what else do we do that stresses mice? Well, we handle them by the tail, and this is for real. It's an easy change for you to make and an easy change for you to advise your investigators to make, but this can be really difficult for your animal care staff who are used to handling animals with forceps or lifting animals by the tail, who are maybe afraid of mice or rats, and so are used to handling them a particular way that gives them a lot of comfort, but that's very stressful for the mice or rats. And handle them at all, but mice and laboratory mice and rats are domesticated animals, so with just a little bit of extra effort on our part, we can make them comfortable with handling and gentle them to the procedures we'd like to do. So hopefully this will start. Yes, okay, so this is a movie, because it's not a talk without a movie, right? This is a movie from Jane Hurst and her work, Taming Anxiety in Laboratory Mice. And these are black 6 mice that have been handled solely by picking up through the tail, with the tail. So you can see how they're interacting with the hand of the investigator. And in just a couple of seconds, you'll see one of the mice, see that? That was defensive burying. So he does not like that hand. Doesn't want anything to do with it, so he tries to bury it in the bedding to be like, no, it's scary, do not want. So when you handle them in a cup or a tunnel or your hand, these are the results you get. And those mice are the opposite of afraid of the hand. I mean, as you can see, the investigators, they're trying to go visit the investigator up on her arm, and they're climbing all over the hand. They have no fear of the hand, no fear of the glove. They're acting like completely different mice when exposed to a human. So we're using this at Harvard with animals, especially some of our wild-caught animals. We use tunnels instead of hands, and the mice train to it exceptionally easily, and we have had great results. It doesn't affect their reproduction or behavior in any negative way, and it makes our staff love it because it makes them easier to change. They don't have to chase animals around with forceps. They don't lose as many animals. The animals don't lose as many animals. Animals don't freak out and jump into we used to have to change them in these deep bins because the animals would be like, wee, and they're peromyscous, so they're kind of wild and very active, and they would try to escape. So this way we don't have to do that anymore, which is better, all the way around. And, everyone wants to tame and train all of the big animals and even rats, it's super cool. But in this case, we had a study where we wanted to inject mice with LPS and study their sickness behavior and how it worked, how it affected nesting. And we found that just handling the naive animals had a similar effect to being injected with LPS in terms of their behavior. They were not normal. So we acclimated them, we shaped the behaviors we wanted, and we acclimated them to all the training, all the behaviors or all the things that we were going to do to them. So this is Brianna training a mouse with chocolate milk, and most of the mice really like the chocolate milk, and she's acclimating him to being scruffed. And it went from being held in the hand to being scruffed to being poked with a dull needle to being poked with a sharp needle to finally the LPS being administered. So handling by the tail is problematic, and there are more and more papers coming out showing validating Jane's work and how important it is. And I really think this is going to be the next sea change, and we're going to have to figure out the biosecurity aspects of that, but I think that we're going to find forceps going away, I hope, or tail handling at all. Lorenzini found that rats unfamiliar with handling really don't enjoy being picked up at all, so whether you grab them, around their chest or back or tummies or by the tail, they didn't like it at all. But rats are really, really smart and good at being gentled. So if this is some of Sylvie's work on rat tickling. There's some other work. There's a Jove video by Brianna and Sylvie, I think, on how to tickle rats properly so that you get the positive results that you want in those 50 kilohertz chirps that they make. And it does, building up these positive interactions with your rats buffers the negative interactions that you may have to have with them. But another cautionary tale from relatively recently is that you have to consider other environmental factors when you are doing this. So animals that are socially housed or animals that are housed in super enriched environments, you may not see the same positive results from tickling, or they may be muted by the fact that rats have these other rewarding things going on. So let's do some more things that stress out our mice and rats. Let's be male. Sorry. But there's enough, overlap between testosterone signaling that male mice being handled by male humans are all like, all right, are we going to fight? Let's go. And they activate that endogenous opioid system, so in case you do decide to pick a fight with them, it will be less painful for them. Be human at all? I mean, again, an apex predator. Do you pet your cat before you leave for work? You're wearing gloves, but still you've got cat dander on you, I bet. Do we feed mice incorrectly? Oh, we feed them so incorrectly, and again, that's another whole long talk. We give them access to the same food all the time. That's a problem. They see into the UV. We don't give them access to UV light because it causes cancer in us and causes problems. We give them lots and lots of bright light so we can see them. They're not really keen on that. We turn the lights on and off, sometimes at random times. We transport them to the testing area, daylight savings time. Although there's a really good, there's a body of work in hamsters because they hibernate, and they use them to study jet lag. And apparently, for daylight savings time, it's a day per hour. So just don't do anything the next day, and you'll be fine. They'll have adjusted to the time change by the next day. And finally, back to the 6 questions, what principles of experimental design and statistics are being ignored when we choose our models? And of course, since Joe wrote this part, and Joe's kind of a statistician, we have a lot of questions here. So we, our scientists and ourselves, we don't know about advanced experimental designs. We assume that all of the important things are controlled because we can measure things, certain things, but are they all important, the things that we can measure? We ignore experimental design and statistics because we don't have a primary hypothesis and a primary outcome, and you can see that with the pressure to publish that investigators feel and p-hacking after the fact to find those significant results, because you can't publish a negative result, whatever. We ignore principles of experimental design because we don't consider our false discovery rate. We don't consider that if our p equals 0.05, well, that means that there's a 1 in 20 chance that was a fake result anyway. So do we consider that when we sort of we said yes, well, this is a thing. We also ignore when we misinterpret confirmatory experiments and technical replicates as bolstering evidence. So just because we can do it again doesn't mean that the first time we did it was right. And we ignore randomization, blinding, and other cornerstones of good experimental design and statistical practice are ignored. So I don't want to get into the weeds on a lot of that stat stuff because I will freely confess that it's like over my head. But randomization and blinding, those I get. So if randomization matters, you almost can't randomize too much. But if I'm unpacking mice and I open the box and I take the first mice that come to hand and I assign all those first mice to the high dose group, what might be different about those mice? Maybe they don't care. Maybe they're the ones that were awake. Maybe they have a different circadian rhythm. Maybe they're more bold in their personality characteristics. How is this going to affect my study going on? So be sure that your scientists understand the difference between random and arbitrary. Right? So arbitrary would be taking all those mice and putting them in the high dose group. Random would be the first 5 mice. Maybe 2 of them go to the high dose group, 1 goes to the low, and the other 2 go to the medium. But I don't know that right now because I haven't generated my sequence of random numbers from random.org to tell me what to do. So a little plug for random.org. It's great. Okay, so what else can we do? Again, as I said earlier, prepare and arrive, and you can register your studies. So if you're doing a human clinical trial, you have to register these studies. You have to register, what you're going to do, your primary hypothesis, your outcomes that you expect, and there's a similar registry for animal-based studies that's out of the EU. So I would strongly encourage you to talk to your scientists about this or to register your own studies ahead of time so that you are clear what your potential outcomes are and you don't indulge in p-hacking and other sort of statistical or experimental manipulations because you expect to see a result or you really thought you would see a result or you wanted to see a result. So what else do you want to do? You're the subject matter. We're lab animal veterinarians, we're the subject matter experts. Speak up, offer help, get involved, collaborate. That's what we're there for, is to be that resource. Be aware of what you don't know. You can't control those unknown unknowns, but I really don't know anything anymore about non-human primates. So if someone asks me about those, I turf that out to a colleague because I would not feel comfortable giving those recommendations or answers. And then keep up with the literature, even if some of it is of questionable validity. That was one of the most trenchant questions I got at one point when I was showing all those papers was, well, how much of this work that you're showing us has been replicated? And I'm like, well, if you look at the body of work, you can see trends, but individual works, I don't know how much of it's been replicated, and I honestly don't know about funding or anything else that's going to allow us to replicate some of this work. So we almost have to see lab animal and husbandry changes as big trends. And accept the challenges that we're given from both within and without the field to give the public confidence in our use of animals in biomedical research and that we are responsible stewards of this huge trust that we've been given. So with that, of course, I'm going to acknowledge the authors of all the works that I cited or showed you pictures of herein, and with mice, we owe a huge debt to Peter Crowcroft, Joe Garner, my colleague there at the bottom, mousebehavior.org, Brianna, also at the bottom. They both look happy because they're on vacation, not at work. And the numerous researchers doing other work on the welfare and census of mice. And then if you're looking for primary sources for rats, John Calhoun, his work is available, it's actually done in Baltimore, in row houses in Baltimore, semi-naturalistic. He explored the behavior of rats in the wild environment. Charlotte Burns, Emily Patterson Kane, Jody Sharp, Sylvie Cloutier, Usama A. Abu Ismail, and then ratbehavior.org, who's Ann Hansen, who's got a nicely curated bunch of stuff online on just explaining basic rat behavior and how they live and work. And so with that, I'm actually finished. Thank you very much for your time and attention.